Okay, friends. So for this part of the lab, we're going to do this part of the lab as a demonstration in class, but just in case you have missed it, then you can watch this video. Basically, this part of the lab is called, it's table 2 on page 3, I believe it is. Yep, table 2 on page 3, and it's asking about the chemical test results. So we have set up our artificial cell, which I forgot over here. We have set this up already, and we are waiting to see what's going to happen. I promise you, hopefully something will happen. But basically, we want to know when we see what happens, we want to know what that means. So the whole point of doing this is to know what it means. We are going to see some changes are going to occur in this beaker. And the trick here is to know, okay, well, what do those changes mean? What does it tell us? What information do we gather from this? Kind of like the boiling water example that I was talking about. So I have set up here 6 test tubes. Excuse me. 6 test tubes, and I have 2 things here. One is called starch indicator solution, which we already used on the outside of our beaker here. That's what's in here. So then you need something called Biuret solution. Okay, and that is kind of, I call it like a glow sticky blue color. It's a light blue color. And then remember that the starch indicator, obviously you can't really see it through the glass container, but it is that amber colored. That's what we describe it as. Okay, so here you can see, where's my mouse? There we go. Here you can see the chart that you're going to fill in as we do this. And again, basically we're just trying to figure out what would happen if and when. So what I have here is 2 test tubes that are for distilled water. One says distilled water with SI, so with starch indicator. This one says distilled water with GI, with glucose indicator, which is what this is. This one is starch with glucose indicator. This one is starch with starch indicator. This one is glucose with glucose indicator. And this one is glucose with starch indicator. So again, I'm just going to switch these so it goes glucose starch each time. Okay, so basically what we want to do is, in order to know if a chemical is present in these types of experiments, we are looking for a color change. So a color change tells us that something is present. You're testing for the presence of something, either starch or glucose. You're testing for the presence of it. We want to know what happens if it is present. What happens? How do we know that that's the case? Oh, my hot plate's hot. I didn't know what that sound was. My hot plate is at temperature, so don't touch it. Okay. So we start with distilled water for both because we want to have a control. We need a control group so that we can compare. So we are looking to see if a color change occurs. Now remember, this one is light blue. This one is an amber color. So if the water in here turns a light blue or an amber color, then that doesn't really count as a color change because that's what color this stuff was to begin with. So it doesn't really count. So the first one here is glucose indicator. I'm actually going to do all the glucose ones first because the glucose is a little bit unique in that it needs to be heated in order for it to work. So I'm going to put a couple of drops of glucose indicator into the distilled water, a couple drops into the starch, and a couple drops into the glucose indicator. So obviously we're testing for the presence of glucose. So only one of these should react that there is glucose in it, and it should be the glucose one. We know it's glucose. So I'm going to take this glucose, this one, and this one. And again, they are all labeled. I know they're probably backwards for you guys, but they are all labeled. And I have back here behind me, you can see over my shoulder, I have it's not boiling, but I have a container of hot water. I'm going to put these in there. A little too much water in there. Okay, I'll put those in there. I'm going to let them sit for a few minutes. In the meantime, I'm going to test these ones. Okay, so starch indicator doesn't need time. It doesn't need to be heated. It will change immediately. But we're going to put starch indicator in distilled water. So hopefully I would not expect to see a color change. Will it change to be this amber color? Yep, sure will. That doesn't really count as a color change because that's the color that it was originally. So this is a negative reaction. So in your data chart for distilled water with starch indicator, you're going to put, I can't see which one, starch indicator's on the bottom. So in this box here, you're going to put a negative reaction, and that it's amber. That's the color. Okay. It's if we put starch indicator into starch solution, the expectation is that you would see a color change. And look at that. You certainly do. So this color change, now there's some starch settled at the very bottom of my test tube that it's not kind of reaching, but if I kind of shake it around a little bit, it probably will. But you can see it's changing to like this black color. Okay, it is no longer that amber color, and it is now a black color. So that is a positive reaction. Usually this is a little bit closer to like a greenish black. It should turn what we call a blue black. It's almost like a really, really deep purple. But this one happens to be just really like black. So for starch with starch indicator, which is this box, you're going to put positive reaction, and that it's black. The last one should be glucose. There should be no starch in the glucose. So we should expect to see no color change other than to turn it to that orangey color. And in fact, that is exactly what happens. We just turn it to that orangey color. Okay? So now we will take a few minutes to let this sucker go. Okay, so now if we take a look at our heated samples, and they're still not quite done, but you can see the results already. Now, I didn't leave these in here for too long, so they're not super hot, but normally they'd be super hot. So distilled water, we should still expect to see no change, right? And in fact, we don't see a change because there shouldn't be any glucose in here. So this should be a negative reaction. And the color, I would call it light blue, even though on the camera you can't tell it's light blue. It is a little bit light blue. So that will be for this one here, top row right there. Then we're going to do right here the glucose indicator with the starch solution. And again, yes, it's cloudy because that's what the starch solution is, cloudy. I'm trying to keep it from dripping on my Chromebook. But it's still just that clear, cloudy, here I am shaking it around, getting it on my Chromebook anyway, color. It has not really had a color change. So that is a negative reaction. And that one is, it does have a slight light blue tint to it. It's not a lot, but a little bit. Okay. And then the last one, which is where we should see the positive reaction because it's glucose with glucose indicator. It was a light blue because that's what color the indicator is. Remember the indicator looks like, go gadget arms. Remember the indicator looks like this. It's that light blue color, okay? So when I put it in, it should have stayed light blue if it was negative. And you can, it's a little hard to see in the camera, but it is turning this orange color. It would not have turned this color if I didn't heat it. It would have stayed that light blue color if I didn't heat it. But it is turning this orangey color because I heated it. And so that here is a positive reaction for glucose, and you'll put that it is orange as well. So each of these 6 boxes should say whether the results were positive or negative and what color the ultimate outcome was of those things. Okay? So that is how you do that, and that is how we did it in class. And now you can see that it's all, should be all filled in as a class we did this. Okay?